burns out. We're like falling stars, miracles dancing on the ground. And we're right with you to feel it try to deep dive. We're right with you, yeah. And we're right with you to feel it try to deep dive. Suffering grows, yeah. You say that life is beautiful. Would you let me in? No, yeah. They say that life is beautiful. Would you let me in? No, yeah.
Welcome, everyone. I have a very, very, very special guest. I would like to introduce you to Dr. Professor Stefano Turini, who is a trailblazer in the realms of molecular medicine, bioinformatics, and nanobiotechnology. He's got a flair for innovation and a passion for pushing boundaries. Stefano's journey from the halls of academia to the forefront of research is nothing short of extraordinary. Dr. Torini's academic journey includes significant contributions to the fields of pharmacology, molecular genetics, spectrochemistry, and as a senior lecturer and research director, his expertise extends to cutting edge research in medical biotechnologies and bioinformatics. And with a prolific publication record and a leadership role in numerous research projects, Dr. Turini continues to pioneer advancements in applied biophysics, molecular dynamics, and innovative therapies, demonstrating a commitment to shaping the future, both through education and innovation. From unraveling the mysteries of molecular dynamics to spearheading groundbreaking projects in medical nanobiotechnology, he's a force to be reckoned with. With a knack for turning scientific curiosity into tangible solutions, Stefano is rewriting the script on what's possible in applied biophysics and beyond. So get ready to be inspired by his visionary approach and relentless pursuit of positive change. And be ready to have your world improved just by listening to Stefano. Stefano, welcome. It is such an honor to have you.
Thank you very much for this opportunity, especially because is the opportunity to express what I have done with my group in cooperation also with some of my colleagues in Italy that as me we like to explore some kind of field of science that somebody can define a fringe science, but I like to define that the real science. Instead of my work that here will be of course expressed also in a part of nanobiotechnology, bioinformatics, the part also which is about the study of the biophysics. You mentioned before also the biophysics. The applied biophysics in this last year, especially the research that we made about the electromagnetic waves and the study about the plants gave to us some kind of the new kind of results that permit to us to remove some kind of the paradigm that we have until now and to explore some other kinds of words that really we couldn't imagine, especially about the results. Some of that published, some of that that are waited published, because we have accumulated what is a real huge amount of data that require different interpretation. And somebody can ask, of course, why until now we still didn't publish all of this data? Because these data are complicated to examine from many different points of view. And I like the presence of the expertise of different experts of different fields that can give their logic, scientific, and technical vision to interpret this data. I don't take care that somebody will say to me, no, this data cannot be interpreted, no, this data cannot include it because the method that you used is etc. etc. I just like to have an opinion. For this reason, in what I will say now and later also, I will give the freedom to the people to decide later, expertise, not expertise, also because not all the people that will listen that, of course, will be expert. But just to give an opportunity to people to discuss about that, to examine that, if there will be some expert between them, better, and to give some kind of answer, because all of that will help us to proceed. One positive answer is one step more. One negative answer is always and again and anyway one step more, because it doesn't exist a positive or negative judgment or positive or negative definition about something, because everything to give some kind of opportunity to go further. In a function of that, we can start with that, and I will be so proud to share with you our work from approximately 2 years and a half ago until now. The titles can be in some mood a little bit provocative. Somebody can say also a little bit with the limit of definition of science. But if there is something that I like is to push the definition and to provocate also. Then the people, they have also to accept a little bit of that, because it's also a mood to give some importance about something that some people only they understand, and less than they can think that can have some kind of future. Entanglement, teleportation, the function and the effect of the sounds about the biological effect. It means molecular music therapy, which is the effect of sound and frequencies at the level of the biological systems. To finish with the part that seems separated but is not, the part about plant neurobiology and behavior. The concept of neurobiology, and I have to say all of the greetings to one person, was introduced by one Italian researcher and vegetal physiologist, Stefano Mancuso, from the University of Florence, that was one of the first people to introduce the concept about the plant neurobiology, to open one kind of vision about some kind of remotion completed, of destroying of the paradigm, smashing really on the ground the paradigm that plant, they cannot communicate if not through the chemical messages. No, they have some other kind of mood of communicating. And here I will show to you also why all of that. I would like to start with one thing before to speak about all of this. All biological systems, and we introduce here one first paradigm in science. All biological systems function with water, that is one inorganic compound. And this is the first paradigm. Biological systems that for definition are organic, they are made for the 80%, like a human body, in example 80, 85, the count is different because the level of the water changes differently and completely during the day, is made by something that is inorganic. How it's possible something like that? And water is a messenger, is a vector. Somebody define water some kind of the living organism. We will not arrive so far, but we will base about some results that were obtained to formulate our conclusions, because the previous research, the experience of the past, is a success for the future, always. I want to citate this publication about some kind of the group of research in which figurate the Nobel Prize and famous Luc Montagnier, and also the other, our national physicist Emilio Del Giudice, recently disappeared, unfortunately. One of the mind with Giuliano Preparata and other scientists like Kamerlingh and Nose, Fleischmann and Pons, the electrochemists, for the formulation of the concept of the cold fusion. Emilio Del Giudice contributed that for the research in a nuclear fusion, control the nuclear fusion but in a form of the cold fusion. They made some kind of the very interesting research. Why they call that DNA waves and water? But first of all, in the references of this, it's possible to see, like also they previous published differently, and it's possible to see directly publication of Montagnier and colleagues, publication of the Judici, Fleischmann, Preparata, Pons, and the others. What they did, even what I will say, it seems incredible on the limit of science, but what they obtained like a result. Luc Montagnier, for many years, they worked, as we know, about the infection of HIV, the human immunodeficiency virus, the etiological agent of AIDS. But he discovered that in many kinds of patients that were infected with AIDS, there was one bacteria that worked in a combination with the HIV virus, Mycoplasma pyrum. And in some other kind of
Wait, you broke it. Wait, hold on. Stefano, can you say that again? That last part
about what?
of what it was found, of the one that was found. Can you say that name again? It just broke up for a second.
About the bacteria, you mean, that was found in HIV, you mean?
Yeah, the bacteria that was found.
Like it's possible to see bacteria here. It's possible in this picture that I'm circulating here. Mycoplasma pyrum is the bacteria that was found in the infection. Mycoplasma pyrum. Mycoplasma are some mycoplasma, like you want to call, are some kind of bacteria that are differentiated with respect to the other because differently respect to other bacteria, mycoplasma, they don't have cell wall. They have cell membrane, but they don't have cell wall. And was found in a combination to work, like to sustain the infection of the HIV. Luc Montagnier and colleagues, they, through some kind of special instruments that they realized, they discover that these bacteria, like also the virus, emit some kind of the frequency of the resonance when it's inside of the aqueous support that's made by the water. Because all of the living organisms, when they move, they emit some kind of the resonance. It's the same principle. Before to speak that the concept of the bioresonance is something that is ascientific or not scientific, it's better to consider also the definition of the nuclear magnetic resonance, because all of the atoms we have, all of the protons, they emit some kind of the frequency of the resonance. And nuclear magnetic resonance function in this mode, orientated the protons with the magnetic momentum inside of one direction until is applied the magnetic field. And when is put the magnetic field in one orientation in 90 degrees and stopped, all of the atoms, they return to the initial position, but in this returning, they like some kind of the elastic movement. And this elastic movement emit one frequency that is detected by the instrument. Molecules, bacteria, viruses, fungi, protozoa, algae, all of the cells in the aqueous part, they emit some kind of the frequency of the resonance that is possible to record. But the question is, what is the effect if I record this frequency of the resonance? That is a train of frequency, because as you can see here, it's not one only frequency, it's not one mono frequency, one own sound, but are many different sounds that is possible to elaborate, to study the signal, to study the spectra, the morphology of the spectra, like we did also with my group, and later I will explain to you better why. Because until now, all of this part seems a little bit confusionary, but be sure that later, maybe in some question that you want already to ask, you will have an answer inside of this. So, to go a little bit further. First of all, how they can detect that? They realize one very simple instrument made by one coil in copper, put inside of some Faraday cage made by mu metal in some insulating metal that they use also inside of the headphones or the diffuser for the music to create some sort of the perfect isolation, connected with one computer. The group of Montagnier and also before, they study also on the basis of the genetical sequence of the bacteria, in this case of the Mycoplasma pyrum, because on the website of the the National Center for Biotechnology Information, part of the NIH of Bethesda, the National Center Institute of Health of Bethesda in Maryland. They have a data bank with all of the genomes, and there is a site also about this, about the Mycoplasma virus. One time that they recorded the frequency of the resonance of the genome for each single base, and somebody can ask, sorry, how it's possible to detect the frequency of the resonance about each single molecule? It's possible. And later, we will discover why, because it's not something that happened in these last years. No, it's something that happened in the years of 1970 through the genius mind of one French scientist and musician. And later I will pronounce the name because the surprise is still to arrive. But they did something of similar, because type of research is similar. They created this apparatus, homemade apparatus, and they created two different lines in this experiment of tubes. Inside of these tubes, they put water with inside the one section of the, we can say like that, of the genome of Mycoplasma pirum, one sequence of the nucleotides of the genome of the Mycoplasma pirum, one specific sequence that they have sequentiated, and they have an example A, T, C, C, G, T, C, C, etc. But they know, was well known the sequence. In the other, they put water, 3 nucleotides, and all the apparatus that is necessary and batteries of enzymes that are necessary to make and to synthesize DNA. DNA polymerase 1, 2, 3, helicase, and G-rase enzymes. These 5 actors, they enter in a synthesis. What they did, they put the tube contain water and these 3 nucleotides inside of one generator of frequency that generate 7 Hertz of frequency. I would like just to make a puntualization that 7 Hertz is the frequency that is near of the frequency of the resonance of the ionosphere, called also Schumann resonance. And I would like also to put one sort of paradigm. Many people, they think that the Schumann resonance is the frequency of the resonance of the planet Earth. Is not. It's the frequency of the resonance of the ionosphere when lightnings are discharged inside of the atmosphere. The frequency of the resonance of the ionosphere, not of the Earth, of course, is reflected on the Earth, but ionosphere and Earth, they have different density, so they vibrate in a different frequency, because the wavelengths of the sound waves, and sound waves, they acquire, it means wavelengths, and they reduce potence when they pass through some substance with a different density. Higher is the density, reduced is the frequency, and much higher is the wavelength. They put inside of the generator, and they mentally that for many hours. And what they understood after that without to make some kind of process, in the tube that there was water and 3 nucleotides treated with the frequency of the 7 Hertz, was assembled one sequence that was the same sequence like there was inside of the other tube. So we can ask, by recording the bioresonance frequency of the genome of specific bacterial, I read what I put, of course, or viral, fungal, or parasitic species, and sending this frequency on a substrate culture medium in the organism, it's possible to infect the later through the frequency only? Practically, we can produce matter just through the sound. We have to say, the physicist and Nobel Prize Max Planck said, the matter exists just because exists the sound capable to put in a resonance the matter. Maybe something he wanted to say through that. And does the single bioresonance frequency of the genome carry within the information necessary to be able to mimic an entire organism? Some kind of difficult question to answer, but we will go to discover better later. In Italy, and this is one master thesis that I found about one military doctor that took, since he developed some kind of the futuristic vision about the application of the research of Montagnier, del Giudice and Preparata, he arrived to think about how this kind of research can be used, since he is a military doctor, he's a military colonel, Stefano Esterino, in a biotechnology applied to the biological war and bioterrorism, new agents as biological weapons. But we know perfectly all of the history about biological weapons. In many books, there is so much literature about that. But he put inside of one part some kind of new agents, especially in this part, experimental theory of the new biological weapons from electromagnetic interference. Practically, he defined all of the research, he took the research, he based himself, he has had an inspiration from the study of Montagnier and colleagues, and he arrived to discover and to say it again, to express again all of the experiments made by Montagnier and colleagues, to describe how they made the experiment, and to arrive at the end to theorize that the future of a biological war will be not anymore the use of the typical biological agents that cultivated in the laboratory, modify genetically to create an hybrid between microorganisms among many different microorganisms, bacteria, viruses. No, the future of the medicine, biological war, he said, could be, between many different things, also the use of the electromagnetic agents, in which will be not anymore possible and only to put some kind of the physical microorganism, but just send the frequency of the resonance of the genome of this specific microorganism. Because the theory that we elaborated, me and my colleagues, if I send the frequency of the resonance of some microorganism inside of the one organism, in this organism, if is an example, the frequency of the bioresonance of some pathogenic bacteria or some pathogenic virus, this organism will develop the disease or symptoms derived from the disease from this bacteria or this virus, even was never put in a direct contact. This is the question that we still don't have an answer, but we have already some kind of the pre-proof, some kind of the first proofs about all of these things. This man, that I wanted to say before, is a man to open one path, is a man to open one road. Joel Steinheimer, one French physicist and musician, that introduced the theory that since that all of the organisms, they vibrate and they emit resonance, it's possible to record that. And it's possible to record that especially by the molecules, each single molecule. What he did, for each single amino acid with his specific apparatus, he detected the frequency of the resonance, and through the consideration and application of the theory of the harmonics in the notes, harmonics are the multiple of the notes. If an example, we have one frequency, an example, just to explain better. If the frequency of the glutamate is an example, the frequency of the bioresonance of the molecule of the glutamate is 4.32 Hertz, some kind of ELF, some kind of the extreme low frequency, we can use the multiple of that, the harmonic, at 432 Hertz. So he understood that each of single amino acids, through the theory and application of the harmonics, they have correspondence with the specific musical notes. Here, another kind of the representation with the diminution for the expert that can define directly the each single notes about that. Here there is one exposition about the Steinheimer work, how he did the publication that he made, also some kind of in French, but that is a demonstration about what he did in his scientific work. And here there is some kind of example about the molecules that he produce the sounds. And just would like, I hope that you can listen also that. First of all, the alcohol dehydrogenase, one enzyme that we have in our patrimony. Etc., etc. Lysozyme, one enzyme that is present inside of our saliva and contribute for the demolition of the polysaccharides in disaccharides and monosaccharides. The sound is a little bit different, but it's possible to appreciate. It's a train of frequency, like you can appreciate. It's not one mono, but one train of frequency. And this is the frequency, like for the alcohol dehydrogenase, this is the sequence of the primary sequence. It means the sequence of the amino acid of the lysozyme. And at the end, the rhodopsin, another molecule that is present also inside of the bacteria, what is called bacterial rhodopsin, and like for the shape of this molecule, is one molecule that interact also and play a role also into the metabolic function in which is involved also the light. As you can hear, there are many different of these sounds associated with the molecules, and each of them they have some specific fingerprint in all of that. So we extend over our research. We arrived to apply the theory of Sternheimer, especially during the COVID pandemic, to understand what is possible to do. It's possible, like we ask to ourselves, produce some kind of the electromagnetic vaccine, and we publish, of course, about that. Just to use the frequency of the bioresonance of the virus put inside of one treated water, some kind of the informatized water, put that inside of Faraday cage to maintain the frequency of the resonance inside, and we develop also some kind of the formula, some kind of physical mathematical formula to calculate how much remain the frequency of the resonance inside of one medium as a water. But before to put inside, we need to put the frequency in the opposite phase. It means not one frequency, but one anti-frequency. To the inversion of phase, it's possible to do by the software. So this is the frequency of the bioresonance of the genome of the virus SARS-CoV-2. Like some kind of the Morse code, etc., etc. This is the frequency of the bioresonance in opposite phase, like in inversion phase. You will not hear probably the difference, but in the biological level, the difference is the opposite respect to what is produced at the level of the cells. Difference there is no, because ear is not capable now to distinguish. But if you put the two frequencies in a modulation in frequency, the amplitude, two same frequencies in the opposite channel, in the opposite phase, to put in the same channel, they erase and produce zero. This is the effect that we want to produce. It means to use some kind of the electromagnetic interference, like appear in the thesis of the colonel medic, the colonel doctor physician Stefano that I mentioned before, to use electromagnetic interference to erase the bioresonance emitted by the virus inside the organism through the use of some anti-frequency. And we publish also about that. We developed, first of all, one software that we called Songenprot Solaris, one software that is capable, based also on the matrices of conversion of Joule-Steinheimer, mathematical matrices that associate musical notes, harmonics with nucleotide ATGC and also U, in a substitution of the theming between DNA and RNA. And we created this kind of software. These are the matrices of Sternheimer with a combination between amino acids, nucleotides, and musical notes with specific harmonics, and send that, because we tried that first of all about the patients, for the reason we call that molecular music therapy, in form of binaural tones. But what are binaural tones? Binaural tones, something that many people maybe they could already have listened in the past, is not something that is possible to emit, to create physically, but is one phenomenon that is generated directly inside of the brain when two frequencies are listening the same time and are with a difference of the frequency between one and the other at least around 30 and 40 hertz. Imagine the same music listened from two different headphones. It's possible to make the experiment. One phone that produces one music, the other phone produces the other music, the same music, but with a difference of 30, 40 hertz. And it's produced one interesting phenomena at the level of the brain. The concept of the binaural tones was introduced in the middle of the 19th century by Heinrich Wilhelm Daube, and discovered, in fact, this phenomena and published on one of the first editorial of the Scientific American that we have also in Italy, this the Italian correspondent of the Scientific American that we call that Le Scienze, and define the nature of the binaural tones and which kind of the effect it can produce at the level of the brain. Physically, and from the point of view of the applied medicine, could be used in this case to make an favor. It means to nourish the production of the estrogen, can have an effect of the Parkinson's disease. Parkinson's disease is associated with the depletion of the dopamine in the level of the organism. Is associated with the metabolism, and it means also with the dopamine in a combination, also in contrast with acetylcholine, and also with other mediators like adrenaline and noradrenaline, the treatment of epilepsy, and hormones also. But the physical procedure that we made inside of the software, how it's possible directly to convert this protein or nucleotide sequence in music? It's very easy. We check one sequence in a bank of NCBI. We copy it in some bioinformatic format inside of inside of the software. We pass inside of that, and software will make the arrangement and to create the effect. This what you listen is the frequency of the bioresonance, the sirtuin-1, derived from the molecule, from the gene, etc., etc. SIRT1. SIRT1 is one gene that produces this molecule sirtuin that is associated with the biological life and with the phenomena of antioxidation and elongation of reduction of the dimension and length of the telomeres. Telomeres are the terminal part that produce and contain terminal part of the chromosomes and contain not codified part of the genome. The length of this is determined by the presence of one enzyme that is called telomerase reverse transcriptase, or better called simple telomerase. Is one retrotranscriptase. Retrotranscriptase is some kind of the enzymes that operate some kind of the opposite function than not the DNA polymerase, not RNA synthetase or the synthesis of the RNA, not from DNA to RNA, but from RNA to DNA. Is some kind of the enzyme that exists in the nature, also inside of the viruses, inside of the HIV, not the case HIV, is called retrovirus lentivirus because it's so slow in the reproduction. In fact, the latent period of that is about 15-20 years, because before to show the first production of the first level of the symptoms. And also one other virus that doesn't have one enzyme that is specifically one retrotranscriptase, but function in a similar mode. In this virus, hepatitis B, is a Pagna virus that has one enzyme that is similar to the retrotranscriptase, even is not specific. We checked on the page of the NCBI. We go on gene because we want some specific gene. We check an example of the human sirtuin-1. We arrive to detect the format FASTA, as I indicate here of that. This is the sequence. We copy the sequence. We put inside of our software, and the result is what I've showed to you now or before a few seconds ago. We apply this kind of frequencies in our patients in a clinic here in Belgrade, in experimentation that we made in an era pre-COVID for more than 1 year and a half. 117 patients treated for some disturbance that arrive from the hypofunction, we can say like that, of some specific protein that derive from some kind of the aberration about some kind of the monogene disease. Disease, it means that derive from the unfunction about only one gene. And to the treated of those with the molecular music therapy applied on one bed that emit also infrared, we show and we know about the effect that was. How we detect the effect? We take blood samples from the patients, we make a genetical analysis of some specific genes that now I will show to you, and the proof that we obtained also we publish later. But why? To use the emission of sounds, it means phonons and infrared and photons. It's not a case that we use that. Like it's not a case that we use the headphones specifically connected with ears. We could put headphones in other part of the body. No, we put it in the middle of the ears because binaural tones produce the first bihemispherical synchronization. The two hemispheres of the brain with the binaural tones that will be in a synchronization. They vibrate on the same frequency. Only through that, the message that we give in a sort of in a form, of the emission of the sounds that correspond to one specific gene sequence will spread inside of the water of the body, and the content of the water of the body will make the effect. But it's not only necessary photons. Photons combined with phonons here and photons, infrared photons emitted, when they interact in some specific heat section, they produce one quasi-particle caused from the physical point of view, called solitons. These solitons, now I will not go deep inside of the physical concept because it's necessary to have a very deep knowledge in physics to understand the nature of the solitons, but it's just necessary to understand, like from this publication that, as we see, they discovered already before of the 1990, like how the solitons, they can interact with the DNA. The emission of the sounds, visible light, or far infrared, like we use, EMF, phonons, means sounds and photons, they interact with the DNA, then produce the effect that we have had. Effect that I said now. The effect to make a vibration on the DNA, and this vibration on specific part of the DNA to this specific fingerprint that derive from the sequence that we send, this has a capacity to induce the expression of the DNA without use any kind of factors. It means only sound can induce modulation of the expression of the DNA. Sound is one of the first factors for the epigenetics. My voice, the sound outside, klaxon of the cars, the trumpet of the cars, the music that we listen, any kind of these things has an effect on our DNA, especially on the behavior. These are the statistical analyses made about and detected by, I made this statistic with one statistical software called the GraphPad Prism. And we can see here before the treatment is the black column, and after the treatment, what happened that in this group of patients that we have had a different group of patients, and some are cancer patients. Cancer is one condition that can derive from the genetics also, but it's not one genetic disease. Cancer, first of all, is not disease, it's a condition. A condition that is established inside of the body when there are no factors like oxygen and right pH. Because cells, they have some kind of the escape waves when the conditions are not good. In a normal condition, cells, they reproduce through the mitosis, somatic cells just through the mitosis, and sperm cells and sexual cells, of course, through the meiosis. But this happens just in a condition of right quantity of the oxygen and right pH, around 7.4. That is a physiological pH. When this condition they have to miss, it means to reduce the tax of the oxygen and is reduced also the pH, become acid. Cells, they change. They cannot make anymore the easy way of reproduction, and they change their status. They change their status in indefinite reproduction and immortality. Immortality is the cancer, because cancer reproduces indefinitely, and cancer reproduces better in the absence of the oxygen and with the low pH. So, in some kind of the primordial earth, at the beginning of the burning of the earth, 3.7 billions of years ago, and later when was developed and start to appear the first ocean, the Panthalassa Ocean, when the waters, they were still rich of acid with a low pH and with a low presence of the oxygen, and also the atmosphere was lack of oxygen, there was a very low level of the oxygen, probably was a word specific for the development of the cancer cell. But we cannot know that. For this reason, cancer in the presence of the oxygen reduces the reproduction, and if we regulate the pH inside of the organism with a good diet, especially to introduce food that give the balance of the pH, buffering of the pH. Buffering of the pH is possible also to obtain by the drinking every day some kind of a glass with sodium bicarbonate and a few drops of lemon. Somebody can say, but lemon is acid. True, lemon is acid, but has also one secret. Lemon can activate better the Krebs cycle, because the Krebs cycle of the tricarboxylic acid cycle starts specifically from the citric acid. And is one way how to make the cell breathing that inside of the mitochondria. So lemon activates mitochondria from this point of view. As we can see here, after the application of the MMT, the molecular music therapy in cancer patients, level of the sirtuin-1 associated with biological life increased. Effect of telomerase increased. It means that there was a difference in modulation of gene expression, and TP53, the most important oncosuppressor of the liver of the organism, with PRB, the protein retinoblastoma. TP53 and PRB are the two most important oncosuppressors. With our MMT, we stimulate P53 to increase and to modulate the effect to stop the indefinite reproduction of cells in a cancer patient. Same thing about the neurological disorder patient. We increase with the MMT sirtuin-1 telomeres after the application of the sequence of sirtuin-1 TP53. And the same about that. Look the results with this big statistical significance that here expressed that is really statistical significance with the application in TP53. Practically rise up that. And this was one of our most important results. Neuropsychiatric patients with depression, with the major disturb of depression, sirtuin-1 is applied, and also values increase, telomeres increase, P53 increase. There was not some kind of big statistical significance, as we can see here, but anyway demonstrated that there was a modulation of the gene expression. Autoimmune disorders like less rheumatoid arthritis, Crohn's disease. As we can see, in one patient that was already applied the biomagnet for the modulation of some specific frequency, as it had already hid the level of the sirtuin, telomeres, and TP53. And this was also a demonstration, a genetical proof, that these biomagnets that are sold in a trade, they have some kind of the effect, they are really some kind of the efficacy. So here we obtain some kind of double results that prove that biomagnet, they have an effect, but also our MMT has an effect. And this was the condensations of the hour fatigue in the article that we published in the Open Access Journal of the Complementary and Alternative Medicine with my group of research, of course. And this publication that with this DOI, this number, you can find on the web, of course, on the PubMed, or on the website directly on the UPIN publishers on the open access of the complementary and alternative medicine. But we went also over of all of this research, not about the complex molecules of the nucleotides of the amino acid, but simple molecules like water, to detect the frequency of the bioresonance of the water, and especially about deuterium, the second isotope, stable isotope of the hydrogen, because hydrogen, for the people that they don't know, they listen us, exists in three different forms, three different isotopes. Prosium, that is the basic hydrogen, with the atom that is composed by one proton and one electron. Deuterium, one proton and one neutron. And tritium, one proton and two neutrons inside. Is radioactive, is a beta emitter, tritium. We recorded the frequency of the bioresonance of deuterium, and we sent in one sample of water all of our experiment we made inside of one Faraday cage to avoid any kind of contamination with the external environment. We made analysis in the Institute of General and Physical Chemistry of Belgrade to one technique that used one nuclear magnetic resonance spectroscopy at this level of the frequency of the magnet is applied. The initial hour samples contain inside 148 ppm of deuterium, and after that we obtained 52.5%. Here there was no conversion between ppm and percentage, but this was an increasing of more than 3,000% of the concentration of deuterium. And this is a proof that sounds that we send inside the nuclear magnetic resonance detected the presence of deuterium. Even we didn't do nothing physically, if not to send the sound, the frequency of the resonance of deuterium. The same in some other kind of experiment, other experiments, in which the percentage was higher in the second experiment, and confirm also that deuterium, that is heavier than hydrogen, is deposited on the bottom of some glass tubes that we used. In fact, on the surface was already high the concentration of deuterium. On the bottom, on the base, was higher on the demonstration that deuterium was created and was generated and deposited on the bottom of this specific water tube that we used. But also other experiments that is in confirmation. Other experiments that confirm how it's possible to obtain many different kinds of the deuterated molecule. An example, we treated in the same kind, not water, but oil, and look the difference of the deuterium treated with the frequency of the bioresonance of deuterium before and after. The difference is practically possible to see, and the results speak by themselves. We use also emozoin, that is one pigment that is released by the parasite of the malaria, mixed with the colloidal silver that we use to try to neutralize the effect of the emozoin. And also other kind of this is one specific protein that we use also for the our research. And as I said, the mozoin is one pigment that is released by the Plasmodium. Plasmodium is the genus of the parasite, etiological agent of malaria, especially the well-known is Plasmodium falciparum. We detected also when we recorded the frequency of the bioresonance of the lithium deuteride, that is the one kind of salt that is used like a compound as a first fuel for the nuclear fusion inside, of course, of the weapons. And at the beginning, we treated with the frequency of the lithium deuteride one tube of water that contains inside this initial concentration that you can see here. After the treatment of this frequency was this 8,500 against 262.
Wow.
Like this, for the oil and this, the results spoke by themselves. And everything is obtained by this nuclear magnetic resonance. It means the direct scientific proof. And we went also over of that. We wanted to prove definitively our theory, and we did the same experiment, but just with hormones of hypothalamus, hypophysis, and thyroid. We recorded and we generated the frequency of the bioresonance of each single hormones, and we have sent that on the tubes of water completely closed, hermetically closed. We send this frequency, we close inside of one Faraday cage, and we put inside of the laboratory of the Institute of General Physical Chemistry for detect the presence of the hormones inside. Why we have chosen this? Because it's rare to find some kind of contaminants in the water like thyroid, hypothalamus, and hypophysis and pituitary gland hormones, because are not common contaminants. For the reason we have chosen that also to avoid all of the possibility to be in error. And after the treatment, what said the instruments revealed the positive answer. It means the presence of thyroid T3, you can see here, T3, T4, PHT, and calcitamine. All of those, they were present inside of the sample of water. From hypothalamus was detected this CRH, GnRH was not detected, GHRH, RH, TRH, and vasopressin. These, they were detected. About the pineal gland, SCTH, FSH, GH, and these, some were they detected with some specific activity. They were not only detected, but they were active compared with the standard that we use for the determination. And all of that just to send some kind of sound on these samples. For this reason, it's possible to make apply of this research for the future, but in many different kinds of future, because they can open many different scenarios that they can define all of these things. There is one researcher, Robert Skopec, in Slovakia, that worked hard to understand how to mitigate the effect of the electromagnetic waves at the level of the organism. And the best is to use for him nafazoline. People maybe they can know nafazoline because it's one compound that enters in a common use like a nose spray also.
Ooh.
This is nafazoline nitrate, and this is nafazoline hydrochloride, but the basic molecule is the same. He detected that there is a mitigation of the Fry effect of the microwaves and other weapons that damage human internal and endogenous organs through the use of nafazoline. What means? The use of nafazoline prevents the effect of the microwaves at the level of the organism. And in 3 different publications, Robert Scott defines specifically that. So, how much compounds like they enter in our common use, they can be useful to protect against about something of sometimes it is really dangerous. 5G emissions is in the range of microwaves. then can produce specific biological effects. And with this, it's possible to mitigate. Like also our publication that we put, and I put also here, that to treat some kind of the depression mood, we used the application of one technique of diagnosis, one kinesiological technique called Orin test. But in the patients, they were treated with one opposite phase frequency that we created, because we detected that in the patient that they suffered by depression, all of those, they vibrate, especially the brain, about some kind of frequency. There is one predominance of some specific waves. By the introduction of the opposite waves, by the listening, the same waves but in opposite phase, remember what I said before. The same waves in opposite phase on the same channel is erased. Our aim was to introduce another frequency to erase the primary frequency that caused depression through the introduction of the same but in opposite phase frequency. And in many of these patients start to decrease the level of depression, and after approximately 7-8 treatment, they start to have again the production, the correct production of serotonin in the body, because one of the main cause of the depression,
serotonin,
even is divided in many different forms, is the inhibition of, is the recaptation of the serotonin in the presynaptic terminal. When serotonin doesn't arrive at the postsynaptic terminal in the synapses, the receptors in the postsynaptic, they cannot have an effect, because it's recaptated at the level of the pre. Antidepressive drugs, they have this effect. They inhibit the recaptation in the presynaptic level, and they permit to the serotonin to arrive in a postsynaptic.
Well, that's the thing, is that that's where, like, treating the parasites, because the parasites are stealing the serotonin, and so that's why it's so important to do that.
Everything, parasites, bacteria, viruses. In fact, there was one man, one researcher, Royal Raymond Rife, that introduced the concept of the resonance frequency, and especially active resonance frequency. And for each kind of microorganism, there is one specific or many specific frequencies, one spectrum of frequencies that is possible to use to induce lysis on this specific microorganism and to treat about that. But not only him, many different researchers and applied researchers, they use this technique for the treatment of parasites, bacteria, fungi, protozoa, that are specific like that. Parasite and protozoa are the same concept. I just wanted to say that. But look at what I mean, because in my movement with the hands, sometimes I cannot express what is the difference between frequencies in phase and dephase, and in opposite phase. When you put some frequency like that, because we discover in our research that people in depression, they have a predominance of the one frequencies in the brain that is around 6.66 Hz. And I don't want to go over why there is this specific frequency. Free interpretation. When we create the opposite phase frequency, you can see here about the peak. What means opposite phase? Is practical like a mirror. All of the mono frequency is the mirror respect of the other. Is the same, but turn in the horizontal plane on 180 degrees. When the two frequencies are summed, there is a modulation in frequency in the amplitude, and there is a tandem repeat of frequencies. When there is the two frequencies are in opposite phase, they sum and they erase. This was our principle into use in this kind of technique that we call OPFT. It means opposite phase frequency treatment. Here I put, but unfortunately it's not possible to hear, or maybe it's possible to hear a little bit the difference respect the 6.66 Hz. Unfortunately, it's not possible. And in the spectrum of the analysis is here. What is not possible to hear, it's possible to see. that. Here is the spectra of 6.66 Hz, as you can see here. To enlarge, it is here on the basic, because it was a pulsative monofrequency. And to enlarge, I just wanted to enlarge it to show to you the band in which it's made, because it was not one pure signal, and this defined it was not one pure signal. Minus 6.66 Hz. Look the shape. You see that it's different? The shape of the spectrum define also and confirm that we could produce in opposite phase both, and to put the sum together, then produce the erasing of that. In phase, counter phase, or in opposite phase, you can see specifically the difference of those, and also about the pulsative. Here there was some time of the division of the each single of peak of that, as you can see perfectly here in the spectral analysis. What we did also, strong of our results, we said, but we can do that also about the microorganisms that normally they degrade the plastic for the reduction of, especially about the microplastic at the level of the sea, and we made, of course, of that some kind of experiment. We produce this specific frequency of the bioresonance to our technique, to be our software, Songer from Solaris, about CDS hydrolyze, MHET-ase, and PET-ases. This was the most important, the last, because it was polyethylene terephthalate ase, PET, is the specific definition of polyethylene terephthalate, one kind of plastic. And this enzyme present in this bacteria is capable to degrade. You can hear also the sounds about this kind of the enzymes. Created by our software. Seems the same because with this speed of reproduction is not possible to understand difference between one base to the other, but software of course can define. And spectra can define also the difference about the zones of density inside of the spectra. Not only that, to produce some kind of the frequency, to produce the opposite phase of frequency about some kind of virus that are very dangerous in Africa and were cause of pandemic in Zaire, in Congo, especially in the last years and after the 21st century, it appears some kind of the Ebola pandemic also in Africa. We produced the genome, of course, of the Bombali, mutant Bombali, Ebola virus, Zaire, Ebola virus, and we put that in opposite phase. This is what we are doing now as a research, and we are collecting data. So since that we are collecting, I cannot still show this our data that we are collecting, but little by little we will publish about that on the scientific paper. Just to listen one sound of that, like appear it seems it is the same like the others that we have examined. Not only that, we worked about the organic compounds like a sort also of the organophosphate like a neurotoxic compound, like this Novichok that was well known about the attempt of the Sergei Skripal that happened by this specific compound. We produce the electromagnetic version about this kind of compounds. We put in opposite phase to erase the frequency of this. As you can see, how much is possible to apply these techniques doesn't have limit of application, and day by day we discover a new level of the application. You can see also that there's a different kind of sound applied. This is to listen in opposite phase, the frequency for the erasing that. And of course, the definition and the application of the applied neurotoxicology about EMF antidotes, because all of these things are EMF, some kind of the electromagnetical antidotes made by the treating of some samples of water with inside the informatization of this frequency, like this kind of molecule, to degrade these organophosphates. The sound, as you can hear, is similar, but to some with the other, they produce one effect decreasing, one erasing of the effect. Moreover, we examine also this kind of bacteria, one of the mostly interesting archaea. It means extranophil of microorganisms. The archaea are microorganisms that are capable to live in very, very extreme conditions inside of the sulfuric source, or in absence of the oxygen, in the ice, in the mud, in the stratosphere, or also in the bottom of the oceans. And they live normally in a condition of temperature and pressure that are impossible to live for the other microorganisms. Thermococcus gamma tolerans is one microorganism that, thanks for these kind of molecules, especially MRE11 and RAD50, especially this molecule, it's very important because permit to this bacteria to resist of the very elevated concentration of the gamma rays, like also the Deinococcus radiodurans, another kind of bacteria that present one kind of the enzymes that are similar about these molecules that are produced, that are also, and I mentioned here. The sounds that you can hear, it means always the same because it's a sequence of the bases of the DNA. We study about the insect, how to modulate also the homeotic gene in the insect for the growth and the development. An example in the flies, into the larvae of the flies, as if you have seen sometimes the larvae, the worm, before that it was developed, the insect, is segmented. This segmentation depends about this homeotic gene, and in each segment of this, there is a different expression of the homeotic gene. For the head, for the thorax, for the wings, for the abdomen, for the arms, for the legs, for the internal organs. And this homeotic gene play an important role in all of that. And we produce also the MF version of that. As you can see, the sound and frequency is of course different because each molecule, each sequence of molecules is also different. We study also the EMF version of some genes of the tardigrades. The organisms with a dimension compared with the acaris, they are capable to resist in a very extreme condition, to slow down the metabolism and to remain latent for decades, for 10, 20, or maybe many years. The secret is inside of the molecule, and we produce the versions of this kind of genes that, as you can hear, has some kind of sounds that is similar about also before. And later also we ask to ourselves, but if we want to go deep in the study to understand how not these deep sounds of the molecules, but sounds that are in our common life, they can have an effect about some kind of disease. If you want to make some kind of the easy, simple therapy, how we can do that? To compare three different kinds of music and sounds that we did, Gregorian, Byzantine, and Tibetan, we ask ourselves which kind of effect that they can have about the reproduction of the virus. In this case, as we can, there's some kind of frequency that can induce relaxation or produce some kind of the effect in some kind of the mystic environment, or the Byzantine. or the guttural sounds derived from the Tibetan chants. Etc. I want to focus your attention about one important point. Look the spectrum, because now will arrive one surprise. And concentrate about this limit. Focus about this number, 14.3 kilohertz. And remember what I said before about Joel Sternheimer. What I said that each part of this presentation are connected and is necessary to understand the part before to have free access and the keys of the kingdom of the next slides and the next argument. Consider this and examine this limit of here when there is the limit of the spectrum. And the mixing of all three. You know which kind of effect with the modulation in frequency and amplitude can produce. The Schumann resonance that we spoke already before. Schumann resonance, the frequency of the resonance of the ionosphere is produced when the lightnings are discharged in the ionosphere, is emitted in three fundamental frequencies: 7.3, 14.1, and 20.3. Do you see something? Do you remember something about before? 14.3, 14.1? Why? These three mystic and derived from the ancient knowledge, music, they have a peak that is around one harmonic of the frequency of the bioresonance, of the resonance of the ionosphere, of the Schumann resonance, 14.1 hertz, and 14.3 or 1 kilohertz. Kilohertz is an harmonic, 1,000 times more than this. In fact, as you can see, all of three, they have the limit around 14.3, very near to this Schumann resonance. Coincidence or something more? The enlargement of that, it's possible to admire that. All of three types of chants have a peak of frequency around 43.3 kilohertz. It means 14,300 hertz. And this is almost exactly the harmonics of the frequencies of the Schumann resonance. All of the harmonic values of 1,000 times higher in frequency than the basic values of the Schumann resonance. It means every time that are made these, there is some kind of the emission in the ionosphere that produce one effect. Of course, many different lightnings, they have a different resonance effect than not some kind of people that they sing that, because effect and intensity is different. This is a typical butterfly effect. One butterfly moved the wings in Peking, and in New York arrived rain and not anymore the sun. And this is some kind also of a mathematical effect derived also from the chaos theory. Is irrelevant. Like when a person swims in the sea, when a person go out from the sea, has some kind of residue of water on the body. The level of the sea is decreased because some part of water is on the body of the person. Is irrelevant,
Yes.
but physically is like that. The effect is the same to understand that. We study. This is the spectrum of the genome of SARS-2. As you can see, maybe it's not possible to see. Maybe I needed to enlarge that. If you can see this point is the highest of the frequency of the SARS. That is near of 14.3. Is not specifically 14.1, but is around. Which kind of these three sounds, Tibetan, Gregorian, or Byzantine, can be the best for that? It's necessary to understand the density of the sound. The best density is again the Gregorian, the similar, basically on the base, and Byzantine. So the application of both, we discovered that they have a positive effect in the people that they are effects of SARS-CoV-2. There's a mixing about this and produce this. With the Byzantine, you can see the mix of both. By the listening, it's not possible to understand because it seems some kind of chaos of sounds. It's necessary to be an expert to examine the specific spectra to understand where there are the point of the erasing, amplification, modulation in frequency and the amplitude of specific sound. And this also with the Tibetan, we applied, of course, the same in the last. What I wanted to say about before that we started our program here, our live streaming.
Mm-hmm.
If people, they don't come in this region of the world, in Serbia, they cannot know about some kind of the history happening specifically during the war. During the war here, there was one group that many people define that a musician group called Grupa Šest Deset Devet. That in Serbian it means Group 69. This group of researchers also, they work about to activate the consciousness of the people, to create some kind of consciousness shield that could protect Serbia from the bombing. It's necessary some kind of the open mind, and not only that, to develop just some kind of this idea. Vladimir Labat, another component of the group, one of those was the physicist and engineer Spaso Yevlaich, unfortunately disappeared a few years ago, in a combination also with other people, but especially Vladimir Labat, later, after the Balkan Wars, continued the work. He created some kind of the homemade musical instrument and created this kind of sounds called in this term that reminds some kind of term referred to the ancient Nordic language, Svarnfal. But this Svarnfal is the ancient Serbian language because in the ancient period, the runic, the language of the runes, appeared in Serbia before the northern north of Europe. So this term probably derived from the ancient Serbian that later population from the north part of Europe taken from this. This is a part of the Serbian history that is a little bit hidden. It is possible to understand only to arrive here. This is one kind of sounds that this man, this Vladimir Labat, this artist developed. With these instruments that he created, the peak is around 14.3 like before, like an harmonic of the frequency of the Schumann. The second, like some kind of a Tibetan bell that is turned on, is activated in this case. Also this in this peak that you listen here, peak that little by little go in a degradation of that because this is the initial peak with one line and later become one band because the sound is generated and like the formation waves in the water when you put one object in the water that it's produce the waves that form one circle, this little by little higher, but with the altitude that is reduced. Here is the same. The peak of sounds is the same than in the water. For this reason, water is the first resonant, the first vector for the making all of the reactions. All of the reactions to the our cells, they happen in the aqueous part. Cytosol is some kind of sol gel solution made by the more over the 80% of water in each of cell. We are made by the water, practically. And all of the reaction happen in the water. Everything that I mentioned until now happen in the water, happen in our organism. And water is the base of everything for all of that. You can see the peak, as I show, is around a little bit higher, but around 14.3 Hertz. And the sum with, like we did also before with Tibetan music, Byzantine and Gregorian music, we did the same also for the treatment with Svarenfall of the people with SARS-CoV-2, with the obtainment also of the good results. And at the end, I wanted to conclude about this kind of our research that we made about the plant neurobiology, to repeat the experiment made by one analyst of CIA that in the years of 1960, he developed one technique to measure the effect of the electrical or bioelectrical transmission in the plants to the use of the polygraph of the lie detector or the true detector, like I didn't want to call that. We published that in the Asian Journal of Basic Science and Research, me and my group here in Serbia. One article called Advanced Software Applied with the Polygraph Technique, useful to interpret electrochemical signals emitted from superior plant gymnosperms during the patterning procedure, with the aim to alter the biophysical state of some vegetable and interpret elaboration through the procedure of reverse engineering, to discover if the plants they have a consciousness we have had before, to try to remove this consciousness. For this reason, we call that reverse engineering. We connected the plant with the electrode of the polygraph, connected with the computer, and we gave to the plants one solution of one hallucinogenic compound, dimethyltryptamine, N-dimethyltryptamine, one alcoholic solution of the 0.9%, and to apply also one machine of Tesla, one Tesla device, a Tesla coil, to give some electrical impulse of the plants of 10,000 volts, to understand what could happen about that. And we ask the direct questions, because voice is a sound. Sound is some waves. When waves they interact with the surface or the skin or the plant, they produce one effect on that. One transduction signal in electromagnetic waves. For this reason, we ask directly that. And at the beginning, there was the answer of the operator, because since that the plant cannot answer, we gave to us answer in a function of the plant. But since that the detector was connected to the plants, was necessary to understand if was alive or undeterminate. Before there was some kind of the phase of understanding, we have to say, through the asking of the question. After the injection in this species plant, Trachyena fragrans, with the solution of dimethyltryptamine, if we compare, because all of these questions necessary to compare in a very deep plant psychology analysis of the clinical folder of that, and in all of this analysis necessary to make one comparison, in the question, do you feel that you have a conscience?
Whoa, you injected DMT into a plant. Wow.
Yeah.
Oh my gosh.
Yeah. And the answer was yes, and was undeterminated. What means? Was near that the plant start to lose when we can call consciousness? There is not a scientific answer about that. And also the question that I put can be not considered so much scientific. But it's necessary to understand how there is the variation of the pathways of transduction signal inside of the plant and biochemical reaction that can induce this kind of answer. And probably the best answer, after the application of the 10,000 or 20,000 volts applied with the Tesla coil without or very, very low intensity to don't produce a big damage of the plants. When we ask, do you feel that you have a conscious, and the answer was yes, instrument was detected as alive, like there was a sort of the losing of the capacity through the application of the electricity. Electricity at the level of the body produces oxidation of the neurotransmitter, can produce, like there was an extra of the electroconvulsive therapy when it was applied in the past year to the treatment of the form of depression or schizophrenia, was capable to produce some kind of the erasing of part of the memory. Maybe can be produced in the same about the plants, since that we can suppose the plant can have some kind of the nervous system, not only directly inside of the structure, but also in the roots. And maybe the real nervous system of the plants depends about all of the roots that are connected between them in many plants that are present in some kind of the green area of one forest. Imagine how many connections there are between the roots of the plants inside of the forest. In the pluvial forest, there are at least 500 billions of trees, approximately, in South America. How many roots they have, this kind of plants? Since that is a pluvial region, there is not so much ground, so the roots are not expanding in a deep, but are expanding in a surface, and they have much more mood to interact between them. A real nervous system between the connection of the roots inside of the ground where the plants are present. Maybe there is the real nervous system, not about each single plant, but the combination between one, the other, the second, in which each plant represents just a node, like one single neuron connected with axons with other neurons, and with dendrites, with our dendrites, with our axons, or with the soma, that is the body of the neuron, to form synapses. Axonics, axosomatic, dendrodendritic, dendrosomatic, and dendroaxon synapses. Here can happen the same with the plants. And we publish. This was the first article that we publish here, that we call it the plants consciousness. And through the interpretation of this data that involve also the study of my colleague that is he's an experimental psychologist in Rome, we are working hard also to produce some kind of interpretation of this kind of results, to develop some kind of the proto-languages that exist between plant and not only humans, but also animal organisms. Here is an example also of the other experiment that we made to treat with radiation of uranium dioxide. And we put also what in the theory that now it will be too long to express about the theory of the organs and the pulsation of the human energy developed by the Wilhelm Reich, but we put one version of the organoids to reestablish the function of the radiations alpha, beta, and gamma, but in this case specifically beta and gamma emitted by the uranium dioxide. Expressed in the color of that, you can see the electrode of the polygraphs, the cage that contain a little quantity of the uranium dioxide put inside the directly of the plants, the electrode data, an organoid put on the base of this. We asked the same questions referred about these plants on the basic question to understand better. You can see the difference of respect about this, because it's not possible, it's not important so much to concentrate about the answer, but the comparison between control and treated answer. This can permit to understand what are the difference. After the treatment.
Wait, what did it say when it said, would you like me to remove it? Because the answer is blocked. Would you like that I remove uranium? What was the answer?
Yeah, because that to remove that. A second. Okay. Because I could see here that if I had one part that covered the screen, so I couldn't read it. Yeah, and yes, the first Yeah, sometimes there are some kind of okay. When I asked in the last question, do you want that I remove uranium for that? And it's just in the first answer, treated with uranium dioxide was yes. And the answer detected by the software was truthful. It means only in the presence of uranium is like a plant has had one sufferance of that. And in this sufferance, there was the will and wish to remove that. After the application of uranium, but the removing of that, the application of the organics to removing the radiations, when we ask, would you like that I will remove organic? In this case, the answer was that we gave was no, because it was logic. That for us, something that can produce a remotion of the removing of something of toxic, we don't want to remove that. The software elaboration was truthful. It means plants has had the positive effect of the organic after of the application of that. It means the correct remotion of the effect. It means plant answer correctly the effect, and the biological effect was determinate. What we can say that in all of these things, as a conclusion of everything, the elaboration provided by our software developed with the artificial intelligence show the results in which vegetables they don't only respond to external stimuli, but also the voice that is one external stimuli, is one physical factor, is one emission of the waves in this case. And through the data that we obtain in this, it will be possible through the interpretation to understand how to develop this kind of language. One language between plants that is not only made by the molecules, but through the emission of the specific frequencies, because as we've spoken until now, everything it can be considered frequencies, because as it was started at the beginning, and the words of Max Planck define perfectly what happened here, matter exists just because exists sound capable to put in vibration this matter. And I hope, this is one wish, of course, that all of these things was a stimuli just to understand, just to have another vision, to take a time to elaborate all of these informations. Not the physical concept, scientific concept, no. But specifically the idea why we did about that. Because before to give a critic on us, in positive or in negative, of course, it's necessary to understand better before why we did that. We did because we wanted to be over some kind of the paradigm of science. We did because we wanted to confirm some experiments that we already did in the past. The experiments of the Luc Montagnier in groups, the experiment of Joel Sternheimer, the experiments of Cliff Baxter as a CIA analyst. We replicated something that already was before with another kind of technology to try to give some scientific proofs that maybe in the past was not possible to give, but now maybe will be possible. And I hope that this will be some kind of stimulation for mine, not only mine, but future colleagues and other scientists to develop some kind of the idea how to perform some other kind of experiments. There was one Indian scientist, contemporaneous of Albert Einstein, Bose, that was one of the first men that worked about a very deep research in electrophysiology on the plants. But many different people, they work on the plants, they produce some kind of the extraordinary effect. You mentioned John Sternheimer, you mentioned Cliff Baxter. We mentioned also one man that now didn't make, Luther Burbank. Luther Burbank, one American man with the extraction of the elementary school, it means basic school, but with one specific gift inside of him. He produced many different versions, many different hybrids of the plants, just with the care and with love that he has had for the plant. An example, in one species of cactus with spine, he just said to the cactus, always to the voice, like we did for our experiments, and like did also Chris Buster. Luther Burbank said, don't worry, you don't need these spines. You don't need these spines because I will protect you from the external enemies. And little by little, the plants start to lose the spine. And now it's one common plant, the cactus without spine. If I remember correctly, is one version of opuntia, if I remember correctly. Maybe no, maybe I'm wrong, but okay. One version of this plant, fat plants without spine, that is possible to find inside of many different houses for the people that they like the fat plants and this kind of exotic plants. But only few people they know that these plants derive specifically from the experience of the Luther Burbank. So all of these specific activities, they have a name to expand the paradigm, not to go over that, because we are scientists and we have to think logically. But of course, fantasy must be on the base of everything. Somebody can say that there is some kind of magic all of that. And I always answer in this mode. If somebody read Arthur C. Clarke can know the third law of Clarke. One technology that is enough advanced, it's practically impossible to distinguish from the magic. And I think that we can say
Wow, that was absolutely amazing. We are so lucky to have heard your presentation. And how do people, if they want to seek the sound therapy treatment, how do they reach you? How does that happen?
you mean if they want to use these sounds, how can they do? You mean like that?
Yes.
First of all, this is some kind of software that we still don't patent it because we still have to understand about it. The treatment with some kind of sounds that can produce some kind of modulation of gene expression is something that must be studied. But we have one citation in all of that. One scientist from South Africa developed some kind of a technique that was a little bit similar about what we did and asked to us to citate us in that and to start experiment with the people with his software and his machine in Johannesburg, in South Africa. And we gave the permission, and for us it was an honor because it means that somebody in another part, of course, far from us, they developed the same theory independently from us, and they developed some kind of things, and maybe now this man start to continue to work on that, but this is not something that is not public. If something want to rise up that has to contact us and to contact me, my mail address, my Facebook address, to ask specifically for that. And we could arrange just to try this kind of sounds. I don't say no, of course. For what reason have I to say no for the people that they want just to use that? Me, same. Until a few months ago, I used daily the listening of the Sirtuin-1 to recharge sometime my body or to do some kind of new effect about the telomerase reverse transcriptase. The only one thing that seems to stop but doesn't stop a little bit that is the can I say is that this software is still not public. Then it's not possible to use that from many people, but we hope to arrive to produce one up from that very soon, because we are already in charge to do that. We have enough scientific proofs, and we are near to arrive at this, how can I say, door to open on this bridge. And one time past this bridge, I think that there will be possible directly to sell that. Yes, yes. With the hopes, of course, that people will accept that, and with the confirmation, other scientific confirmation that we have to obtain, of course.
There's been so many, like everything that I'm into and that I preach, I always talk about sound alchemy, that there's a way to harmonize ourselves and to thrive and to live longer by through sound, and that the ancients knew this, and they knew what notes into play in what order, and treating it through frequency. And I speak a lot about parasites and cleansing from parasites, and I see a rise in fungal infection, and just so turning to healing modalities and knowledge and solutions in that. And so much of what you are doing and focusing on, and you discussed, are all solutions to that and studies that can help us. So I'm super grateful. And it was so interesting, and there's so many synchronicities talking to you. Like, it was Walter Bosley had asked me to do a show on the Tsul princess, and I wanted to dive into the fluid. I wanted to dive of like what this potential fluid could be. And you did not know about this, and you happened to message me about this sarcophagus and this fluid that she was into without knowing what
Yes.
I was focusing and researching on. Like, so it was so divine, like it was not an accident. And you really helped me with sending me information about amniotic fluid, vat fluid, immortality of suspended animation. And so there's just, you have such a plethora of knowledge, and it's such an honor to be able to know you. And I feel so lucky, and I'm so grateful to be able to, yeah, know you and listen to you.
What you said is practically the definition of entanglement, because my presentation started with entanglement, but I wanted to arrive to this point because you understood perfectly what I explained, and you arrived to the basic conclusion. The entanglement is a connection of the distance, even since that there is no connection. The entanglement is the physical factor that can connect people on the extreme distance. One mother will be always connected with the sun. Even one is on the Earth and the sun is one interplanetary mission on Pluto, because there will be always one strength, that one force will connect them. Synchronicity, entanglement are the same thing. And you close this presentation with the definition of entanglement. So I think that you couldn't close in a better mood if knowing what you already done. It means you close with the words to speak about the thistle princess, to speak about the coffin, about the water, about the fluid that's present inside, with the content that today I've read your post on Facebook in which I put also the like about the content of magnesium and the other substances. Probably there was a fluid for the preservation of that, since that the study the structure of the mummy from the time before Christ to now, of course, there was glycerin, there was mercury, there was arsenic, all of the things that can maintain and can preserve that inside, because chemically it's possible to use like this. Also, during the emperors of China, when one imperator that now I forgot the name, and I forgot also who, for which dynasty, asked to the air alchemist, I have to live all of the life. I have to live in eternal, because one life is not enough to do whatever I have to do. They answer to him, Magistry, there are two moods, how to prolong the life. I don't know if are eternal, but they can prolong, of course. One of these was the ingestion of the metallic and liquid mercury, of course, in some specific concentration. Somebody can say, but mercury is neurotoxic. Everything can be toxic. It's not, to citate also Paracelsus, is not the type that make the poison, but is the quantity of it. Also the water is toxic in some concentration. Everything depends about the substance, of course. And here is the same. Everything in a right concentration is useful. Everything. Somebody, but no, there are some kind of junk food and everything. Doesn't exist, in my point of view, junk food or something. No. Everything taken in the right mood, and to put ourselves in the right mood to accept an example of this food can be useful. Of course, if you go over that in positive and in negative, produce a negative effect, of course. But it's the basic of the function of our physiology.
Wow, Stefano, thank you so much for everything. I hope to do more shows with you and projects. And it truly was an honor. And just thank you. Grazie.
Prego. Because really, this opportunity that you gave me here today is one proof that synchronicity and entanglement exist. Also about what we have spoken about the project of our book together. This is another type of synchronicity that we will do. And tomorrow, finally, I will start with the index about that. After all the commitment that I have had these days, tomorrow I can finally start with that. That will be much more free to do that. And of course, we will have other meetings about that with other arguments. Because this part is they just still want 10% about what there is. We have to still to develop also the others. And we will develop.
Yeah, it's almost overwhelming.
A great interest.
It's almost overwhelming. Like there's the things that I'm into and focus on and am passionate about is very specific. And it just so happens that you happen to be focusing on like the exact same thing. So it's almost scary of like, oh my God, listening to your presentation, it just absolutely lights me up. Like I just want to smile and cheer. Like, no way.
I'm so happy. I'm so happy to give to you these sensations. And another time, this is an example of synchronicity, entanglement, and to proceed about that, we'll discover maybe that we are astral brothers, like I discovered the time with when my best friend that is in Milan, is also one of my colleague, also with you, maybe we'll discover to be astral brothers, astral brother and sister of that. That we share the same interest, the same mood, how to make and to develop this kind of interest. This is not something that is necessary to take like somebody take like a coincidence. It's not a coincidence. It's much, much more.
Well, once again, thank you very much, everyone. Thank you for being here. This just makes my heart sing. I could like tear up. This was so amazing. And I look forward to hearing from you again and everything that will come. And yes, everybody have an amazing night and arrivederci, Stefano.
Beautiful till it all burns out. We're like falling stars, miracles dancing on the ground. And we're right with you to feel it strike deep tight. we're right with you. Yeah. And we're right with you to feel this time deep down. Suffering grows, yeah. Let's say that life is beautiful.